Hi, hello, my dear students. This is your Prasanna Harikrishna sir, Assistant Professor of Botany, SRR Government Arts and Science College, Karimnagar. So today we are going to see about C4 cycle. Already we have gone through the basic information regarding C4 plants in the last session. Okay, in this session we will see how the cycle takes place in C4 plants. Clear? So this C4 cycle has an alternative names. Those are pep cycle. Okay, pep cycle. Why it is called as pep cycle means this particular C4 cycle has the first stable compound in the name of pyruvic pep. So phosphoenol pyruvate, the first stable compound in C4 cycle is phosphoenol pyruvate. Okay, hence this cycle is also called as pep cycle. Then this is also called as what? Hatch and slack cycle. Okay, so Hatch and slack cycle. These two scientists belong to Australia. So Hatch and Slack, they have done a great research on sugarcane by using carbon 14 isotope. Okay, so these two scientists had contributed a lot in the field of C4 cycle on sugarcane by the usage of carbon 14 isotope. Okay, so hence these contribution of these two scientists is tremendous, and it is awesome. Hence this cycle is also called as Hatch and Slack cycle. Then beta carboxylation cycle. Beta carboxylation cycle. So there are compounds like OAA, oxaloacetic acids, then aspartic acids, then MA stands for malic acid. So these three compounds contain 4 carbons in them. Okay, so these 3 or 4 carbon compounds are oxaloacetic acids, aspartic acid and malic acid. So these 3 compounds have 2 carboxylic acids in the structure. 2 carboxylic acids, nothing but COOH groups. These 3 compounds are having 2 carboxylic acid groups. Hence they are beta carboxylation cycle. Okay. Now, how this particular C4 cycle is represented in the form of diagram. Just have a look. This is, already I have told you how the mesophyll cell and the bundle sheath cell look like. These two are the cells. Okay. So this is mesophyll cell. So mesophyll cell. This is what? Bundle sheath cell. Okay. So this is bundle sheath cell. This is mesophyll cell. Now this mesophyll cells, this is what? CO2. So CO2 enters inside the mesophyll cells. Clear? There is a compound called PEP. So PEP is basically a 3 carbon compound. Okay, so this is the entry of CO2. CO2 is converted into HCO3 minus, that is bicarbonate. Okay, this bicarbonate is used and there is a formation of OAA, that is oxaloacetic acids, right? So oxaloacetic acid, this is 4 carbon compound. Okay, this oxaloacetic acid is converted into aspartic acid. Remember, see, I will draw the diagrammatic representation in detail, right? So this is PEP. So phosphoenol pyruvate, this is a 3 carbon compound. Okay, CO2 enters and it is converted into HCO3 minus. This is useful for the formation of OAA, that is 4 carbon compounds. Clear? Then this OAA may be converted, so enters into the chloroplast. See, this is the chloroplast. So it enters inside the chloroplast and there will be formation of 2 compounds. They may be formed, they may be changed into aspartic acids. It may be changed into malic acid. This is also 4 carbon compound. It is also a 4 carbon compound. Okay. So if oxaloacetic acid is changed into malic acid, there is something happens, that is NADPH is converted into NADP plus. Okay. The enzyme here is malic dehydrogenase. So here aspartic amino transferase. Okay. Now this malic acid enters into where? Bundle sheath cell. This is the malic acids, 4 carbon compounds, and there is a formation of pyruvic acids. So pyruvic acid is a 3 carbon compound. Then what happens here? 1 carbon dioxide. Okay. So 1 carbon dioxide is lost. So this carbon dioxide is involved in C3 cycle. And so due to C3 cycle, there is a formation of sugar. Clear? The enzyme for this particular reaction is here NADP plus is converted into NADPH, and the enzyme will be NADP malic enzyme. Okay. So NADP malic enzyme. Now this pyruvic acid again enters into this particular mesophyll cell. Then pyruvic acid is in turn converted into phosphoenol pyruvate. So here ATP is converted into ADP. The enzyme is pyruvic dikinase. Okay. So pyruvic dikinase. So this is all about diagrammatic representation of C4 cycle. Clear? Just have a look. So in exams, in competitive exams or in academic exams, you can differentiate 2 cells clearly. This is what? Mesophyll cell. This is what? Bundle sheath cells. I have already told you the complete information and the complete differences between mesophyll cell and the bundle sheath cell in my previous session. Clear? So now this is what? Carbon dioxide. Carbon dioxide enters inside the mesophyll cell through the stomata. Okay. Now carbon dioxide is converted into bicarbonate. Okay. This particular reaction is catalyzed by carboxylase enzyme. Okay. Now this bicarbonate contains 1 single carbon. This 1 single carbon associates with 3 carbon compound, that is PEP, phosphoenol pyruvate. Okay. So 3 carbon plus 1 carbon. So 3 plus 1 gives rise to 4 carbon compound, that should be oxaloacetic acid. Okay. So this is a first stable compound in C4 cycle. This first stable compound is having 4 carbons. Hence this cycle is also called as C4 cycle. The first compound which is involved in C4 cycle is PEP. Hence it is also called as PEP cycle. Clear? Now this particular combination bicarbonate and PEP is catalyzed by pepcase. The enzyme is pepcase, phosphoenol pyruvate case. Pepcase is an enzyme which catalyzes the combination of bicarbonate and phosphoenol pyruvates, means one carbon associates with 3 carbon compound, hence there is a formation of 4 carbon compound, that should be oxaloacetic acids. Clear? This entire reaction takes place in the cytoplasm of mesophyll cell. Remember, cytoplasm of mesophyll cell. Now, this oxaloacetic acid enters into the chloroplast of mesophyll cell. Okay, so then when it enters into chloroplast of mesophyll cell, it may be converted into aspartic acids. Aspartic acid, that is also 4 carbon compound, in the presence of aspartic aminotransferase. Aspartic aminotransferase. Or it may be converted into malic acid, that is also a 4 carbon compound, but this particular conversion is catalyzed by malic dehydrogenase. Okay, so whenever dehydrogenase, D nothing but removal of hydrogens or protons. Okay, so NADPH, the protons are removed from the NADPH and NADPH is converted into NADP. Clear? Now, this malic acid is produced where? Chloroplast of mesophyll cell. Now, this malic acid is entered into the bundle sheath cell. Okay, bundle sheath cell. This malic acid is a 4 carbon compound basically. Malic acid is converted into pyruvic acid. This is a 3 carbon compound. Okay, what happened here? 4 carbon compound is converted into a 3 carbon compound, means 1 carbon is lost. 1 carbon is lost in the form of carbon dioxide, means it is purely a decarboxylation reaction. This is what addition of carbon dioxide takes place in the first step. This is what carboxylation, or you can say carbon fixation. Okay, but in this case carbon dioxide is removed from malic acids. Clear? So this kind of reaction can be called as decarboxylation. Okay, now this carbon dioxide enters into C3 cycle and there is a formation of glucose, or you can say sugar. Clear? Now in the conversion of this particular malic acid into pyruvic acids, there is change of NADP plus into NADPH. Okay, protons are added to NADP, then NADP is converted into NADPH. This entire equation or entire process or entire reaction is catalyzed by NADP malic enzyme. Okay, so NADP malic enzyme catalyzes this particular reaction. Now this pyruvic acid again entered back into mesophyll cell, that is cytoplasm of mesophyll cell. Now this is 3 carbon compound. Pyruvic acid is converted into again 3 carbon compound, that is phosphoenol pyruvate. So in this conversion ATP is converted into ADP and phosphates. This reaction is catalyzed by pyruvic dikinase. Okay, so this is all about C4 cycle. Clear? So this is a C4 cycle. In the next session or in the next class, we will see the CAM cycle, crussulacean acid metabolism. Okay, that is also a beautiful cycle. We will discuss everything related to that particular CAM cycle. Thank you, one and all.